Welcome to Library Seminars, NOAA Central Library's webinar series for the presentation of research and ideas that reflect the National Oceanic and Atmospheric Administration's mission. This seminar is part of the Knauss Lunch and Learn series, which is a monthly webinar where the 2022 Knauss Fellows showcase their very own research or the projects they're supporting during their fellowship. Today's presentation, Happy as a Clam, Ecology and Photosymbiosis of Giant Clams in Palau, will be presented by Pennsylvania Sea Grant Knauss Fellow, Lincoln Rehm. Giselle Schmitz, a 2022 Knauss Fellow working in NOAA Fisheries Office of Science and Technology, will be our host and will introduce our speaker. So before I turn this webinar over to our speakers, here are just a few tips to help you enjoy the presentation. If you're having trouble with the audio or visual components of GoToWebinar, please just log out and rejoin us. This will usually reset the software and resolve most of our technical issues. The presentation is being recorded and will be available on the NOAA Central Library YouTube channel later today. I'll go ahead and put the link in the channel in the chat box as soon as I finish talking. And most important, we highly encourage you to ask questions, which the speaker will address at the end of his presentation. So type your questions throughout the seminar as they come to you. You can do this by going into the control panel and putting it into the questions chat box. It should be on the right side of your screen. So I'll go ahead and turn this presentation over to Giselle. The mic is yours. Thank you.
Thank you. So I am delighted today to introduce my friend, Lincoln Rehm. Lincoln is a current PhD candidate in environmental science at Drexel University and is presently placed in NOAA's Office of Science and Technology as a Data Governance and Policy Fellow. His research focus is on the conservation and photobiology of the giant clam. And before beginning grad school, Lincoln worked in Palau International Coral Reef Center as a researcher, studying the effects of climate change and conservation and nearshore resources. Outside of work, Lincoln enjoys a great alliterative list of hobbies: bouldering, basketball, and board games. So we're all excited to hear from you today, Lincoln.
Thank you. I laughed when my title was introduced for this presentation because everyone has had such clever titles for their talks. It took me way too long to figure out that happy as a clam is a phrase that people use. But I'll be talking to you about 2 chapters of my PhD in a little bit about Palau too, which I really miss and I'm hoping to travel back to soon. There we go. Before I start this presentation, of course, I want to acknowledge all the folks who made this work possible. Of course, the Palau International Coral Reef Center was integral in this research. I spent years working there and I continue to build my relationship with them through my PhD, and I'm incredibly thankful to those folks who helped me. The research was funded by NSF, and I worked both with Drexel and Yale folks in the Allison Sweeney lab. And of course, I have to thank my family because I used photos of them in this presentation with their permission to add some context to some of this work with the giant clams. Today I'll be introducing giant clams, I'll be talking about Palau a little bit, and then I'll talk about giant clams in Palau. And then I'll finish off with giant clam photosymbiosis. So where is Palau? Palau is located in Micronesia, which is a region of islands that are located just across the equator and a little bit north of the equator. Palau specifically is located due east of the Philippines and just north of Indonesia. It was colonized by the Germans, then the Spanish, then the Japanese, then the Americans, and then finally gaining its independence in 1994 from America, while still holding a close relationship with them through the Compact Free Association Agreement. Palau is unique in that it has a democratic government with a president, governors of different states. Some of these states are labeled here like Koror, Ngarad, Niwal. But in addition to sort of the democratic government that we know of, they also have this traditional system, which also holds seats within that Senate or legislator. Palau is a matriarchal society where women are voting in some of these chiefs. And of course, if the chiefs are not doing a good job, they can kick them out. So really, it's the girls who run the world in Palau. Palau is most famous, though, for its 300 small rock islands, which form a beautiful coral reef archipelago. When I give a presentation of Palau, I always feel like I have to include pretty photos. So here's some photos that I took, both of my research when I was researching corals in Palau. Don't worry, I'm not touching the corals there, but then also of some of the things that tourists come to see. Jellyfish Lake, the high diversity of fish and corals, and we have sharks as well. Since Palau's diversity is so high, and there's sort of this beautiful natural underwater world, tourism actually makes up 80% of the total GDP. I didn't mention this, but Palau's population is near 30,000 people. It's very small. However, over the past decade or so, tourism has increased to points where there were over 100,000 tourists coming to dive, snorkel, and fish in Palau's coral reefs. Due to that, Palau has experienced lots of stress on its infrastructure and then its fishing environment due to this increase in tourism. That's a little bit about Palau. I just wanted to sort of ground you in where we are. So Palau is actually PW, that's where we are. And this is a map of the animal that I study, the giant clam, and its biogeography. They're typically across the Indo-Pacific and the Red Sea, where some species are only found. In total, across the world, there are 12 species of giant clams. However, in Palau, Palau has 8 species across 2 genera, Tridacninae and Hippopus. These are some photos of those giant clams. These clams can range anywhere from 10 centimeters in shell length, so that's about the size of maybe your middle finger, to over a meter in shell length. And if you remember that photo of me with my arms spread out, that's the same species. These guys can grow pretty big, and they typically live in shallow coral reefs. And you might have noticed there's that photosymbiosis word that was in my presentation, and that's really what enables these clams to live in these shallow, sometimes low nutrient environments, is that photosymbiosis, which I'll talk about later. In Palau, giant clams hold an important cultural significance. In some cases, they represent money. So this is a diagram drawn from this buy house. This is an important meeting house. You'll see giant clams iconography across many of these meeting houses. And in fact, since I study giant clams, I really like this image. I decided, might as well get the tattoo because I'm dorky enough to do that. But Palau also has a legend about the origin of Latmikaik. So Palau describes this giant clam that sat on top of a hill or a mountain in Palau. And one day this giant clam was shaken. Something disturbed this clam and it opened up. When it opened up, out poured all the sea life that we see across Palau. And of course, this important goddess Latmikaik that is really important to Palauan people. So in addition to that cultural significance, giant clams hold socioeconomic significance in the form of food security. So this is my aunt, or my 2 aunts, and this is a picture of what it would look like when they harvested. They picked up some sea grapes here, some sea cucumber, but then right here is the giant clam mantle tissue that many people in Palau eat. It is kind of a staple food for Palauans. But as globalization increased, so did the demand for some of these really ornate animals. So aquaculture and shell trade became really important industries throughout the world, and especially in Palau. This is a photo of a giant clam aquaculture facility that's one of the largest in the world. And these are shells that are sold to international aquariums or even shell industries. So with the high demand of these giant clams for both local food and then international trade, Palau has worked hard to conserve these giant clams. In fact, they've banned the harvest of these for over 10 years. I put 2 asterisks there because I mentioned that my aunts still harvest them. Well, Palauans can harvest these giant clams for food. However, wild clams in Palau are banned from export. This follows a lot of sort of international trade laws where many other countries have instituted similar policies, banning the international export of their wild stocks. CITES recognizes these giant clams. However, their status ranges from least concern, where there's not really much conservation initiatives needed, to vulnerable. I should mention that there have been recent petitions because of the recent influx in population data showing that giant clam populations across the world are suffering. So people have been petitioning to raise their status from most of them being vulnerable to some of them even being endangered. Palau's been in the news as well. I put this photo of a New York Times article for some of the more, I would say, radical approaches to dealing with international illegal fishing and illegal harvesting. This is a photo of a boat that was caught illegally harvesting fish in clam shells. And in fact, Palau took it out to the middle of the ocean and burned the boats to sort of send a message. However, despite all this conservation effort to protect giant clams and other fish across Palau, giant clams do not have a modern baseline of their populations in Palau. There have been surveys in the past. However, those surveys were very specific to and localized to 2 small reefs. So the goal of one of my chapters is to just simply survey clams across the Palauan archipelago and establish sort of a baseline, a snapshot of what do the populations of giant clams look like in Palau. Again, this is a map of the country. The black indicates the islands. The gray are the fringing reefs, and those are reefs that are adjacent to these islands. The blue are these barrier reefs, and the orange are both an atoll and an oceanic reef. We also have pink and green dots, and those are our survey sites. We tried to take an approach where we surveyed broadly across Palau's archipelago, and again, this is just a snapshot of what Palau's giant clam populations look like. So onto the data. On the right is a sort of key for what these abbreviations are. TC is Tridacninae, maxima, NOAA, squamosa, dorasa, gigas, and then HH is hippopus. Sounds like a Harry Potter spell. I highlighted Tridacninae because the work that I did was, or the paper that we published was the first to actually find Tridacninae in Palau. Of course, finding new species is really important to conservation efforts in Palau, as it enables people to really identify the critical needs of species that may be rarer than others. On the left is a figure where the top panel is the density of giant clams, these different species, and then on the bottom is their shell lengths. We compared their density and shell lengths across those 3 major habitat types. Across shell lengths, most of the species were fairly similar, or the densities were mostly similar, sorry. However, Tridacninae, this panel right here, the density was significantly higher in the fringing reef, which kind of follows most patterns that we see in the literature today. What's really cool is that Tridacninae densities are significantly higher than most populations that have been surveyed across the world. So Palau really has an abundant population of these clams. However, the clams are pretty small. When we compare the average size of the most popular species, that would be Tridacninae, Tridacninae maxima, and Tridacninae squamosa, they are significantly smaller than populations found throughout the world. So we wanted to take a little bit of a deeper dive into the population structure of those 3 species. Again, Tridacna maxima follows this column, Crocea squamosa, and then these rows are the reef types: atoll, barrier, and fringing. And what we found across all three of these populations is that the clams lack these adult individuals. This dotted line represents the size at maturity for these three species, and you can see most of these population trends peter out. In addition, giant Tridacna crocea lacks really important recruit individuals. What this tells us is that there's maybe something going on that's affecting these populations, especially the big ones. And if you imagine people harvesting these for food, of course, they're targeting the larger individuals. In addition to the work that we've been doing, we wanted to look at whether conservation status played any role in actually protecting these animals. The white boxes are within a conservation area. The grayed out boxes are outside of conservation area. And again, density is the top panel, shell length is the bottom. What we found is that conservation did not play a significant role in affecting or even positively affecting the densities of clams. Our expectation was that we would have more clams within a conservation area. In addition, shell length really varied across most of these species. And in fact, in some species, we found larger clams outside of the conservation area than inside. We haven't yet come up with a really good explanation for that, but we're still sort of exploring this this result. So in total of this survey, we found that despite Palau's best efforts to effectively conserve these giant clams, they're kind of struggling. We saw a lack of recruits and adults, which indicates some sort of outside pressure, most likely harvesting. And without those recruits or adults, major stressors like El Niños or hurricanes or typhoons can really dramatically affect these populations and even cause a collapse in these species. If we take a look back at the giant clam, though, we wanted to look at these colors. If you noticed in the previous slides, they're really pretty. I'm not sure if this one is hard to see or not, but you can see this is the siphonal mantle tissue. The clams are always oriented where they're sort of settled on the bottom and facing towards the sunlight. And when we take a closer look, we see this siphonal mantle tissue is made up of really bright, I mean, this looks like space kind of, really bright cells and then really dark patches of cells. When we take a cross section, those bright cells sort of layer the top, and those are called iridocytes. And then within the tissue, those dark patches were actually pillars of algae. And we can see that if we sort of turn this scale bar, this algae is actually really deep in the tissue, sometimes 3 millimeters deep within that siphonal mantle tissue. That algae is actually with the dinoflagellates found in the family Symbiodiniaceae. Recently, this phylogeny was redone. Most of the species were under the genus Symbiodinium, but in fact, recent work has sort of separated these clades into their own genus. These I'll call them zooxanthellae for now. These zooxanthellae are commonly found in corals, and they form symbiotic relationships with them, and they do the same thing with clams. They're found in other mollusks as well, and found in symbiotic relationships with other animals. This is an example of sort of the feeding behavior within a coral. Of course, they have the tentacles which come out at night. Those tentacles can then feed on zooplankton, but most of the coral nutrient needs or carbon needs are met by the zooxanthellae. The zooxanthellae sit right under sort of an epithelial layer of tissue, and they undergo photosynthesis. And of course, in return of providing that carbon, they get the nutrients, water, and carbon dioxide that they need to undergo photosynthesis and sort of operate. This is the same thing that happens in the giant clam. However, the giant clam and the coral have come up with two different strategies, very different strategies for absorbing light. If we take a look at this image, corals have all kinds of morphologies. We have tabular shapes, we have encrusting shapes, we have branching patterns, and these are really ways to increase and maximize the surface area that is exposed to sunlight. That way you're absorbing as much sunlight and undergoing as much photosynthesis as possible. If we look at these two giant clams in the middle, they have this giant shell, and that creates a problem for them. How do you increase and sort of maximize photosynthetic production within a clam tissue that's this small when you can't increase the surface area really? Well, it turns out that we look at these shiny cells called iridocytes. Those iridocytes, when you take a cross section of them, kind of look like this. This is a schematic of one, sorry. Within the cell would be a small nucleus, and then most of the cell would be sort of squished or made up of protein platelets. And that's what these little lines are representing. What those protein platelets do is they create a Bragg reflector. What that means is incoming irradiance, incoming light, hits these platelets and just scatters at a slight angle. That scattering occurs throughout the tissue, but what you have is some light scattering forwards. That would be this sort of green when we look at this panel. I think it's panel B, sorry, my webcam's covering it. Panel B, we see greens and some sort of dark reds and some blues scattering forward. But then at these angles, we see that really blues and reds are scattering. And then what we saw in the images were backscattering of greens and yellows, sometimes blues as well. What's really cool about the iridocyte and its scattering potential is that scattering pattern actually follows the absorption curve of the Symbiodiniaceae, or the zooxanthellae. Where there's lots of absorption in the blue wavelengths, there's lots of scattering. Where there's lots of absorption in the red wavelengths, there's lots of scattering. So if we kind of take a very simplified view of the giant clam, again, with those pillars going down deep into the tissue and the surface of them being covered by iridocytes, those iridocytes are scattering those really important wavelengths deep within the tissue. When we model a giant clam with and without iridocytes, we found something really cool. For example, this blue and red lines right here represent a clam without iridocytes. What happens is the light becomes quenched within relatively shallow depths of the tissue. And that's because of the algae absorbing the light, the zooxanthellae absorbing light for photosynthesis. However, within a clam that has iridocytes, that light actually doesn't become quenched deep and pretty much until it hits the really thick muscle tissue of that mantle. So what happens is, So what happens is we have a clam who's really maximized their volume in order to absorb that sunlight, and they have created a system where algae deep within the tissue can absorb sunlight and undergo photosynthesis. If we take a look at the giant clams again, you'll remember that these have really interesting patterns. Remember, this is a backscattering of the light. So we see sort of stripy colors, blues, spots, and these are 3 different species. What we wanted to look at was whether there's some sort of color distribution or patterning across Palau. And this is important because giant clams in the aquaculture and aquarium industry are really targeted because of their color. Some aquariums want blues, some want more of this purple or yellow. And then to sort of expand upon our surveys, we created or we simulated environmental conditions that may impact photosynthesis and, for example, may reflect potential climate change effects on those giant clams. So we did a photo transect survey across Palau at those same sites. Here's an example of what a raw image would look like. We have our color card, and we use that color card to color correct these images. And this is what the image would look like. So we go from really bright and easy to tell stripes to some of these other individuals just being really dark and kind of just brown and yellow. Well, it turns out when we surveyed the clams across Palau, we found that they were a mix of yellow, blues, and greens. And when we take a look at the most abundant species, again, Crocea maxima and squamosa, they actually have a specific color pattern that's associated with those clams, with the sort of extreme example being squamosa made up of almost no blue, mostly greens and yellows, Crocea being made up of lots of blue, and sort of maxima being an intermediate. When we are surveying these clams, we noticed that there may be a pattern in the color and the reef type that it's associated with. So we wanted to test whether that that pattern is real. And so we simulated sort of reef conditions in the lab. We have a factorial design where we tested the effects of water and light treatments. So we have clams that are exposed to high light. So these are really shallow. This is an example of what the tanks would look like. And then one where the clams are shaded. And then we adjusted the water to where we added nutrients to simulate sort of eutrophication. And then again, shade and eutrophication. And I put this skull and crossbones here because unfortunately, working abroad through your PhD means that you have things that happen that are out of your control. And we had sort of a catastrophic power outage at the aquarium, which caused the pumps to go out. So this treatment just died. In addition to the photo surveys that we're doing, we're also using micro CT to take a closer look at whether these light and water conditions impact the morphology of these algal pillars. So if you remember, the algae are oriented in sort of these pillar formations. This is what an example of a raw image from the CT scanner would look like from the side view and then from the surface. So what did we find? Well, we found that light actually plays a significant role in the color morphology of these clams. In fact, over time, it appears that clams became less blue when exposed to more light. When they become less blue, they most likely become more green, and that's what we saw in this image where the dots are 2020, the triangles are the 2017. In addition, we did see some change in the yellow hue. However, they remained relatively stable above that 30% mark. And that's 30% of the tissue being yellow. What's really cool also is that we found a difference in these two treatments: clams that are exposed to high light and high nutrients, and clams that are exposed to shade and no nutrients had major differences, in fact, significant differences in the amount of green hue. The green iridocyte is really interesting, as we're really unable to explain its role, or in fact, what it even looks like. It's really hard to target that green iridocyte and model that within some of our simulations, which are in some of my other chapters that you can ask about later. Another thing that we measured in our experiment is albedo. Albedo is the measure of how much light is reflected from the clam. So the higher the number, the more light that's reflected. Ideally, you want a low number because that means that you're absorbing more light. And we can see in 2017, albedo across these treatments was relatively similar. However, it appears that nutrients significantly affect the clam's ability to absorb nutrients, or to absorb light, I'm sorry. And this could be to the influx of nutrients actually adding or supplementing the clam's nutrition, maybe not relying on photosynthesis. However, more likely is that some of those eutrophic conditions are actually impacting the photosymbionts, or those zooxanthellae within the siphonal tissue. What I want everyone to recognize, though, is that this is 0.1, which means that 90% of the light is actually absorbed by the clams, which is really high. When we look at corals, corals are probably around the 70%, I believe, mark. So despite some of these shifts that we see in albedo, the clams are absorbing tons of light and they're using it to grow. Here's an example of a reconstruction of the CT images that I took of the algae. Some of these look a little messy, but you can see some of these pillars. It's a little easier to tell in this. What we found in our micro CT data is that the morphologies did not differ among the treatments over time. So that would be the size of them, the length of them, and even the way they cluster did not shift across those treatments. So we think that the iridocytes are really sort of mitigating a lot of the light changes that may happen. They may actually shade them and prevent the clam from really shifting their algae within the tissue. So we found that altered light nutrients could possibly negatively impact the clam survival. We saw that some clams were not absorbing as much light. In addition to these high nutrients, typically in eutrophic conditions, you find an increase in disease. And in fact, you see that across some of the surveys that you do in Palau, with some bleaching occurring in environments that are not necessarily hot or high light, but most likely due to some other stress. Despite some of these effects, the giant clam holobiont is really extremely highly efficient at harvesting light. So much so that we've sort of expanded our research into sort of manufacturing synthetic iridocytes to really take advantage of the way that those things scatter. That is for another talk. The main takeaways of this presentation, though, are that giant clams in Palau need conservation intervention. After I wrote this paper, I helped review some legislation that I'm not sure what the status is, but they are attempting to really change the way conservation of the giant clams is done in Palau. And I'm really proud of what Palau is doing. I think that they're really forward thinking. In addition, climate change conditions such as increased light or increased runoff for eutrophication can impact the clam coloration, but more importantly, will increase their survivability by decreasing their photosynthetic efficiency. This was only a piece of my thesis. I have 2 other chapters that I did not present. So if you want more information about giant clams, please reach out to me. Or if you have any questions, you can email me or message me on Twitter. Thank you.
Thank you, Lincoln. That was fabulous. Very interesting and quite beautiful to look at those clams. So audience, we're going to go ahead and take the next few minutes to answer your questions. If you have them, and I hope you do, please type them in the questions chat box and I will read them to Lincoln. And before I do that, I also want to let you know that Lincoln has made his slides available to us. So if you're interested in those, this is the best time to download them from the handouts menu in the control panel. And again, we have been recording this webinar. So if you think that you know somebody who would be interested in Lincoln's work, please feel free to connect them with the recording, which will be on the YouTube channel for NOAA Central Library. And I'll also put that in the chat. So Lincoln, we did have a couple of questions. Let me go ahead and read the first one to you. The first one asks, do the biggest individuals reproduce more? For some fish species, there's a drive to not harvest the biggest individuals. Are there any size limits to the local harvesting?
Palau's only harvesting regulations are for the export of giant clams. So that's true with spawning things too, with especially mollusks. The larger the individual, the more gametes they're producing. So one of the things that we really are really pushing for, because it has worked in other islands, is to place size bands on some species of clams, especially those really big ones, which take a really long time to grow. What I didn't mention is that in one of my papers, we look at the age structure of these clams. What you find is that for a clam to grow to something like 20 centimeters, and this is the Crocea species, I think it takes over 15 years from what I remember. And that may be wrong, but it is a long time, especially when you have constant stress and these acute stressors happening in Palau.
Thank you. This next question asks, does light ever become damaging to the clam? I'm especially wondering about the ones that grow in very shallow water. Maybe they have pigments that screen them from UV damage?
That's something that another lab. So there's the giant clam world is actually kind of small. And another lab is looking at the UV pigments within the siphonal tissue. One thing that I should mention is that these iridocyte cells, not only do they scatter, but they perform like a shading function. You no longer have maximum intensity of sunlight hitting that surface. That being said, giant clams similarly react to highlight like corals do when they bleach. You'll see the clams expel these zooxanthellae and just become totally white. And eventually, for the most part, that leads to mortality, but for some of these clams, they're able to survive.
Wonderful. So you have a compliment and multiple questions from this next person. It says, really enjoyed your talk. Thanks. 1, how have the conservation areas been in place long enough for the clams to grow to maturity? 2, are the conservation areas in places where historically there was relatively high harvesting pressure? And 3, do you have information on how well the conservation restrictions are respected?
That is the last one is tough. I think that especially small islands really struggle in enforcing conservation. However, I think Palau does a relatively good job. Most of these conservation areas are adjacent to the dock or adjacent to the island. So a lot of times they can stand there with binoculars and see who it is. And of course, Palau is a very small country. So if you see someone, you know their boat and you probably know who it is. Let's see, the first question about conservation. Okay, or the two questions. So giant clams, a lot of these protected areas that I surveyed have been in place since the early 2000s or even the late 90s. Those conservation areas were selected, some, I would say some for the clam harvest, especially those seagrass habitats, but then others for just conserving popular fishing areas. There is one survey site that we surveyed that was specifically placed in a popular clam spot. And what we did find was a really interesting sort of bimodal population structure for that clam, where we actually found almost no juveniles or sort of that mature individuals, but we saw or not huge, but large populations of recruits and adults. And we think that could be due to the conservation initiatives in Palau and specifically targeting those clam sites. But with the other conservation areas, I think they're taking a more holistic approach and just conserving popular sites for fishing and harvesting other, a variety of animals.
Thank you. Good memory too for remembering all the questions. Another question. It's a compliment. Great talk, Lincoln. Very timely too, as we in NMFS PR are currently working on a status review for 7 of these species to determine if their status warrants listing as threatened or endangered under the ESA. I'm particularly interested in your recent abundance data, and I'm wondering if the data is published yet, or if not, would you be willing to share? Happy to follow up with an email.
It's published. It's that number 3. So email me as well. I can give you the publication because I think it's behind, it's science, so it's behind some sort of paywall. But yes, I would be extremely happy to talk to you about, whoever this is, about the conservation status of the giant clams, because I think that there's been, like I said, an influx of data, and especially for some of these rarer species, that the status is it's old. They need to update it because some of these clams are really struggling right now.
And just so that anyone else knows, I do share all the questions and contact information with Lincoln, so he'll be able to reach out to you. Next question asks, what are the distinguishing characteristics of T. NOAA? Or again, I'm going to mispronounce this. NOAE.
So I wonder if I can go. Well, okay. Let's see. It's going to be too far. Tridacninae looks very, very similar to Tridacnina maxima. They both sort of bore into coral reefs and grow similar. They're similarly sized. There are shell differences that you can find, and I think that one of them is the number of, oh man, I'm just blanking on the name, the number of the curves in the shell. But really what a major distinguishing characteristic of Tridacninae is siphonal mantle pattern. The mantle pattern of the NOAA is so unique and it looks like teardrop shapes that you just cannot miss it. But you can also notice that the shape of the shell, in addition to sort of the curviness of the wave form, I just can't remember. the shape of the actual shell, the valve looks a little bit different from maxima as well. But it's something that I think conservation managers have to train themselves on because they've just sort of listed that as maxima in Palau, whereas now we know it's a different species and it's a pretty rare one too.
So interesting. There's 2 more questions here. This next one asks, are the clams growing more slowly because of ocean acidification?
Yes, probably. I mean, so ocean acidification really, it really impacts some of these smaller species, these boring clams, because they bore into the almost concrete. And what it does is it affects the calcification of the shell. And so I think what you actually have happening is smaller clams in acidic environments and also really fragile skeletons and or fragile shells, I'm sorry. And this ends up affecting them and increasing their likelihood of becoming sick from other diseases.
Okay, thank you. And this last question asks, could restorative aquaculture play a role in the recovery of giant clams?
I think so. I think that you have it can play a part. I think that like aquaculture in Palau is really working, and especially when you take the approach that Palau does, where they are telling people like farm these clams, you know, have your own aquaculture because it is a source of revenue. We'll provide the seeds, we'll help you with exporting these clams. They really do help the individual maintain their clam farms, and it sort of mitigates the pressures on wild clams. Like that being said, there's still a certain amount of human pressure that's still happening. We have illegal poaching, but then we have the regular harvest of clams, and that really affects them, as you can see from some of the data that we have published. So I think that aquaculture plus a combination of stricter regulations really would positively impact those clams.
Excellent. Thank you so much, Lincoln, for your presentation and fielding all those fabulous questions from the audience.
Yeah, thank you.
I hope you have a great year this year at NOAA, and I hope that people reach out to you. And Giselle, thank you for hosting as well as all the work you do to lead the Knauss Lunch and Learn webinar committee. Audience, please join NOAA Central Library again for a Knauss Lunch and Learn webinar. We host them the 3rd Thursday of the month at 12 p.m. Eastern Standard Time, and learn more about the research of this really fabulous group of fellows. So enjoy the rest of your day, and thank you very much.